Okay. All right.
So hang on a second. Let me.
Okay. Okay. All right. Good morning, everyone. Can people hear me?
Yes, we can.
Okay. Can any of the participants? Okay, I think they are automatically muted. So good morning, everyone, and welcome to the next in the series of webinars under the Kubrick umbrella. Just that this time we also have a sponsor or a presenter or producer of this webinar. That is the Immunological Society of Ghana. So you are all welcome. We have a presentation that will be coming to us. Let us all listen carefully, and then we will have some Q&A afterwards. If you have any questions when the presentation is ongoing, you can quickly use the Q&A window to pose your questions. And then at the end, if you would want to ask a questions live, we can also take that, but I would read out the questions that are posted in the Q&A session. So this morning, we are very privileged to have a good colleague and a friend of mine in the person of Dr. Bright Edu with the Noguchi Memorial Institute, the Department of Immunology. Dr. Edu also is the person in charge of the Noguchi Memorial Institute's Sequencing Hub, which is an Africa CDC Center of Excellence for Genomic Sequencing. They've done a lot of work before or during the COVID. They were there before the COVID, but did a lot of work during the COVID time and are carrying on even now that COVID has gone down. So we thought it good to invite him to just tell us a bit of what applications we can put genomic sequencing into when it comes to disease surveillance and how that supports public health. So Dr. Edu is a senior research fellow, like I said, in the Department of Immunology, and he will be a resource person for this webinar session. So I welcome him to start his presentation, and then afterwards we'll have some Q&A. Dr. Edu, the floor is yours.
Thank you so much, Professor Kusi. I hope I'm coming through loud enough. Can you hear me clearly?
Very, very loud and very clear.
Perfect. Thank you, and for inviting me to do this, and also a big welcome to everyone. So we're going to talk about application of genomics to public health, and we will take it at the infectious disease surveillance angle. This will be the things I would be sharing with you. I want to take us briefly through memory lane or history, a little bit on method development, where we started from and where we've come to, and then discuss briefly some of the leading technologies that are driving genomics globally. The list here is not exhaustive. Just pick ones, those that are a little bit more common, and 454 for historical reasons. And then I will talk briefly about how we have responded to 3 different outbreaks using genomics as a tool to help inform public health. So this will be the outline for the conversation we are about to have. A very busy slide to start with, but bear with me. I picked this from Eurofins Genomics. Can you hear me?
Dr. Edu, yeah, so
yes.
The slides are not changing. We are still looking at the
slides are not
slide.
changing. Oh my.
Right.
Why is that? Should I unshare and share again?
I think that might help, yes.
Let's try. Where is my Let me know if you are seeing my screen.
Yes.
Okay.
Yeah.
And then is this the slide is not moving. Is the slide moving?
Yes. Now it is.
Okay, good. So
Okay.
this is the outline of the talk. We will take a brief path through history. We will talk also about the sequencing platforms, and then I will share with you how genomics has been a great tool in responding to 3 different pandemics, disease outbreaks. I become used to the word pandemic. This is the slide I was talking about. This here shows us a brief history of sequencing, and ever since Friedrich Mischer was able to isolate DNA from the nucleus and show that, I mean, people had already shown that this was relevant for hereditary traits. And when he isolated that, there was always the interest in trying to understand it more. And there was no better way to understand it than to be able to read it in a form that we can appreciate the content or the context of it. And so we all know about Watson and Crick when they proposed the double helix model using data from Rosalind Franklin and others. And that kind of set the pace for so many other very, very landmark molecular biology work. First of which was the sequencing of transfer RNA for the alanine amino acid. And there was actually a Nobel Prize awarded to Robert Hawley for this work that he did. And again, that the methods that were used at that time were mainly analytical chemistry methods. So people were using very dangerous chemicals to try and digest DNA and to look for the way that the nucleotides are stitched together to form a sequence. It is said that Robert Hawley and his team, they had to use about 140 kilograms of yeast to be able to isolate pure enough tRNA from the yeast cell to be able to sequence it. So you can imagine the volume of work these people did, very, very crude techniques. And so a bit fast forward, people began to attempt to do some form of sequencing that is a little bit more less technical, less chemical, it uses less poisonous chemicals. But it wasn't until 1975 that Frederick Sanger came in with a very ingenious method to be able to read the sequence of nucleotides in a DNA molecule. And he called it chain terminating sequencing. And the idea is that if you have DNA molecule, I'll talk a little bit more about it, but just bear with me that you have DNA molecule. It has is a D-ribose oxynucleotide, and it has a ribose sugar as part of the backbone. This ribose sugar needs a certain configuration of H and hydroxyl groups for you to be able to polymerize the chain and make the DNA longer. He found a way to get rid of one of the OHs at position 3 so that he can terminate the chain and be able to read whatever has been produced at any given time. And that became the basis for what we now call Sanger sequencing technology. And it became quite popular for over 40 years. In fact, Sanger sequencing is still very, very relevant today and very, very useful to the extent that even deep sequencing data is often verified using Sanger sequencing. But so people have tried over the years to sequence different things and never been able to get to a whole genome level. Sanger sequencing was automated around 1987 by Leroy Hood and his team, where they capitalized on a very neat technique using capillary electrophoresis. And then in 1995, we had a first bacterial genome sequenced. Now, this was a landmark achievement because prior to this, nobody had been able to sequence a whole genome of any organism. And so this was the first one. Now, subsequent to that, we've had some major breakthroughs. Pyro sequencing became available around just after a year or so, and then that became the first kind of next generation sequences that were available for people to use. And with that, you could go in very, very considerably higher sequencing lengths. So through history, people have attempted to, improve on the technology, and we have gone on and on and on, and we are now having techniques that are now termed third generation techniques, which allow us to sequence longer fragments of DNA with relatively shorter time and also very minimal resources. So the key ones so far are the 4x4 systems, Illumina systems, and Oxford and Paul systems, which I will talk a little bit more about in detail. Now, Frederick Sanger saw this. He saw what we are living in today, what I call the golden age of genomics, because he was able to predict that if we can sequence an organism, we would understand life better, essentially. And this was way back in 1980. And as I said, over the years, a lot of effort and research have gone into improving the technology of sequencing. And this has correspondingly resulted in an exponential increase in the number of genomes that have been deposited in genome databases. Here you see the plot of number of genome sequences that have been deposited in GenBank and the years. Now, what is remarkable, I'm going to switch a bit to my screen. So I'm still looking at you, but just bear with me. I'm going on a bigger screen. What is remarkable here is when you look at the first sequence, the first genome that was sequenced around 1995, the Haemophilus influenzae, it took us quite a number of years before we got about 1,000 genomes in the database. But from the time we got 1,000 genomes to the time we had 1,000 E. coli. So this is, first one was just general, all genomes, doesn't matter what organism. But after that was achieved, in just 4 years, E. coli alone, there were over 1,000 genomes available. Now, just 3 years down the line, E. coli genomes had gone up to 100,000 genomes. And currently, with all the different platforms that are available, PacBio system, Oxford Nanopore system, Illumina systems, there are over 300 million genomes or sequences that have been deposited in GenBank alone. So that tells you that the improvement in the technology has actually brought about a lot of innovation and data available to us. But it hasn't always been that easy. As I said, people have been working for decades trying to read the sequence of DNA. And here we have Alan Maxam and Walter Gilbert, who were using what we call chemical degradation. So they were using very, very poisonous chemicals like DMSO, hydrazine, and others to degrade DNA at specific nucleotide positions. And then when they have a phosphate, which is hot, a radioactive phosphate at the initial 5' position, they are able to run this on a gel and expose the gel to X-ray film so that they can print wherever the band occurs. And by reading this in reverse, knowing which tube contains which digestion, they are able to reconstruct the sequence. This obviously would only give you very short sequences, and it takes a lot of time. You are exposed to dangerous chemicals, and it wasn't the most friendliest methods available. And this was when Sanger came onto the scene. Like I said, this is the nucleotide bases that are polymerizing in DNA. And here we have an H at this 3' position. This H should have been an OH. This is the normal deoxynucleotide. However, when there is an H at this position, there is no longer a chance for this chain to be extended. And Sanger capitalized on this when he introduced a system where he can radio label the D-deoxynucleotide triphosphate and have 4 different solutions with all the conditions that will allow for DNA polymerization. But for each of these 4 tubes, he would introduce just one type of the dideoxy nucleotide. And what that means is that if you have a DNA chain and you denature it by heat and you attach a primer to it, and you start to extend it, depending on which nucleotide is incorporated during the extension, if it's a normal DNTP, the chain will extend. But if it is one of the tagged ones, which is radiolabeled and it doesn't have the OH at the 3' position, the chain will terminate. And so by running these in separate tubes, the incorporation is random. So you're going to generate different fragments with different sizes. And then if you denature that into single strands, you can run this on a gel and they will separate out based on their sizes. The smallest will fall faster to the bottom and the biggest will stay at the top. And so you can go back and read the sequence again in reverse and be able to reconstruct your sequence. This became the basis for Sanger sequencing. But of course, it was required, it was time consuming, labor intensive, and very expensive. You couldn't scale this to sequence multiple genomes or bigger genomes or genes, because the whole process is very, very labor intensive. And the biggest of all, this is extremely low throughput. You can only sequence one target at a time. And so there was the need for automation. And Karamolis had invented PCR that won him a Nobel Prize in, I think, 1985 or so. And this was one of the earliest models of PCR machine. Prior to that, you could have this kind of system where, so PCR can be done basically if you have controlled water baths that you can monitor the temperatures to a certain degree and control that. Because the whole thing is just thermal cycling. You move from one temperature to the other and to the other, and just keeping it at a specific time, you're able to polymerize DNA with the right conditions. So this innovation was then capitalized on by the Sanger developers, and people like Leroy Hood were able to introduce automation to it. So in this case, instead of using radio-labeled ddNTPs, they introduced fluorescently labeled ddNTPs. And instead of doing these in 4 different tubes, you are doing the whole reaction in one tube, because each of the ddNTP has a different color, and therefore there is no interference. And they will all be competing with the normal DNTPs during the polymerization. So you have your templates, you have your primer, you have your ddNTPs, and you have your polymerase. When these are subjected to the cycling conditions, there's going to be elongation. And every time a normal DNTP gets incorporated into the growing chain, the chain will be extended. But if one of the ddNTPs that is fluorescently labeled gets incorporated, the chain just terminates. And that becomes the terminal length of that fragment. And when this has gone on for a while, you can run that in a capillary electrophoresis, and by using laser to excite your dyes, you are able to record the particular strand that is passing or that is available, and the particular nucleotide that is at that portion of the fragment. That whole information is converted to what we call chromatogram, and you can then translate this into your sequence. So this is one of the earliest models of automated Sanger sequencing machines. But again, like I said, these Sanger sequencing is really good, but at best it is low throughput. So the innovation to do massively parallel sequencing was still necessary. And the first breakthrough came through this 454 pyrrole sequencing, which requires that during the incorporation of nucleotides into a growing chain, there is always a release of a pyrophosphate. That pyrophosphate, it gets converted to ATP by phosphorylase enzyme, by sulforylase enzyme, and that ATP is used up by luciferase to convert luciferin to oxyluciferin. That reaction often results, or not often, that reaction results in the release of light. So the number of pyrophosphate released corresponds directly to the number of nucleotides that are incorporated. And this is done one at a time. So the chain is being polymerized, and one nucleotide is flooded onto the flow cell at a time. And when the incorporation occurs, pyrophosphate is released, the light is released, and the signal is detected as an incorporation of that particular nucleotide that was flooded. If nothing occurs, there's no light emission, and the next one is flooded on, the next nucleotide is flooded on, and then it goes on and on and on. There is an enzyme called apirase that is recycling any unused DNTP which didn't get incorporated. So this is the technology for pyrosequencing, and it became very, very dominant in the early 2000s, late 1990s, and it was very, very helpful. It's still in use in most labs, actually. The next technology was the ion torrent technology, and ion torrent is exactly what it is. The whole sequencing technology is based on measurement of protons when there is an incorporation of nucleotides into a growing chain. The number of protons that are released is equivalent to the number of nucleotides that are incorporated. So here, for example, on this growing chain is the same reaction that is happening. You have polymerase, you have DNTPs, but when there is an incorporation of a nucleotide, let's say this is an A, it gets flooded onto the flow cell. The proton that is released is captured by the sensor. This is happening in a microtiter well that has a sensor at the bottom. And this sensor detects how much proton is released. So this one, for example, only one proton was released. And therefore, it tells you that there is just a single adenine that has been incorporated into the growing chain. Now, take this example where there are two incorporations of thiamine. The signal that is released is from these two proton release is bigger, and therefore the software is able to tell you that there's two thiamine that have been incorporated. And this then is also very useful because it's a different technology, but it allows us to sequence bigger genomes and much more at a much more higher throughput. The next technology, which is now one of the main dominant ones, is the Illumina technology, which is based on the principle of sequencing by synthesis. So you prepare your library. I'm not going to go into the details of library preparation, but suffice to say that libraries come in as double-stranded DNA, having adapters attached to their ends. These adapters have complementary sequences to a flow cell, and therefore, when you denature the library and you flood your flow cell with these the library, they get recognized by their corresponding sequences, and they stick onto them like what we have here. Now, the sequencing is done by these nucleotides that are fluorescently tagged. When they get incorporated and the correct one is incorporated, light is shone onto the flow cell, and there is an emission of light. So laser shines onto the light, excites the fluorescent dye onto the flow cell, excites the fluorescent dye, and then this light is emitted and it is captured by a high-resolution camera that is overlooking the flow cell. That information can easily then be translated by the software into the nucleotides, because only one nucleotide gets incorporated at a time, and then the signal is captured. The last one that I want to talk about is the also nanopore technologies, nanopore sequencing. And this is basically having a nanopore, which is a protein pore that is inserted into a membrane that has a very high resistance to electrical current. And another ionic current is established in the pore, such that when you bring a DNA molecule and it passes through the pore, every DNA molecule is it carries a certain every nucleotide in the DNA molecule carries a certain native electrical charge by being a molecule. I mean, it's a molecule, so it has electron clouds, it has atoms, and these charges interfere with the stable current field, and that interference can be specifically measured, and that measurement can easily be translated into the sequence that is passing. So, for example, adenine would distort the current field by a specific signature that is different from guanine or cytosine or thymine. And this is then, the software is able to then use that information to tell us what nucleotide has just passed through this stable this electrical field. That is the information that is translated into nucleotides. Okay. So what are the strengths and weaknesses or the downsides to this? These are a lot of text, but I just want you to focus on a few things. This is another technology that I didn't talk much about, but it's also in the market. It's a PacBio system. Now, all these have different rates of accuracy, and the cost implications per billion bases also differ. And so when you have an outbreak, you want to go in for something that allows you to get the biggest bang for your buck, as they say. And there are different reasons. Some of these machines are very expensive, but they may give you a lot more data. But others are cheap and may give you smaller data. Take, for example, Sanger sequencing. The machine is not as expensive as, say, the Illumina machines, but the cost of 1 billion base pair is quite a lot compared to the Illumina platform. And this depends a lot on the flow cell you are running, the capacity of the cartridge you are using, the size of the genome you are doing, and how much depth you want to go. But suffice it to say that all these technologies have certain strengths and weaknesses. And for example, the nanopore cost, the MinION costs just about $1,000, but your throughput may not be as high as, say, NovaSeq or NSSeq or one of these Illumina platforms. And also the accuracy issues. So this is how the technology stands at the moment. This is how the field looks like. And so I want to now shift attention briefly and start talking about the what happens when there is an outbreak. During an outbreak, there may be an organism that is causing this outbreak. Of course, there is an organism. And it starts off with its own genome. And so let's call that parental strain genome. In the transmission of that organism, as it spreads from person to person, there's going to be, it needs to multiply to sustain the spread. Now, this multiplication requires that it must copy its genome so that the progenies will look like the parent. Every time it copies the genome, they think of the genome as the life book of the organism. And this life book contains all the information about how the organism will behave, what kind of drugs it will be resistant to, how it will evade the immune system or not, what kind of spread, the intensity of the disease, everything. So every time it copies this information, there are likely to be small errors. If the errors are favorable, they get passed on. If they are not so favorable, they get wiped out. So with time, you see that the transmission gets dominated by the most favorable variant or So when we go in, I'm using COVID here as an example. We go in and take a sample, we bring it to the lab, we prepare it, we do the library as we discussed, and then you run it on the machine and you do some kind of bioinformatics to get your data. Now, why do we do this? Why is genomic surveillance so important? Because we can use that to track the movement of pathogens. We can track them over vast areas of geography, and we can use this data to also help us develop interventions such as vaccines and drugs and diagnostics. By understanding this, we can also check the effectiveness of our interventions. Are our interventions working? If it's working, how does it affect the population of the pathogen that we are trying to stop? And we can do all that. Now, the mutations that the organism accrue, as I said, may translate into fitness in terms of transmission. For example, what we saw with the spike gene mutations in SARS-CoV-2, or it could cause severity in the disease, or it could cause it to evade the immune system, or it could make it resistant to diagnosis, like what we have for HRP2 and HRP3 deletion in Plasmodium falciparum. So what are we actually looking for when we get a sequence? We're trying to establish relatedness between the different sequences. Take these 5 sequences, for example. they all share this particular mutation. And these dots are like mutation. But 3 of them share this mutation. Only 2 share this. And therefore, we can then bring in something that makes it a bit easier for us to relate to them, what we often call phylogenetic trees. Now, these trees, so you can see these 2, they are more closely related, so they sit on the same branch, and that is why you have them here. So in an outbreak, you can see, oh, okay, maybe all these 3 have an ancestor which has this mutation, and therefore, this is why they are all sitting on this kind of branch here. Then all of the organisms have this common ancestor, and therefore they share this branch, and they are all, before they got splitted into these. But when we look at phylogenetic trees, we often have a lot more information than we have just looked at with this dummy. Phylogenetic trees can contain information. So for example, all these here are sitting on this here. That means they are more closely related. And often, if we know a bit about the origin or the start of the transmission, we can root the tree and we can put a time scale on it that allows us to understand how the organism is changing with time. Here, for example, the length of this is a time optimized tree, and the length of this here tells you something about how much time has elapsed since the original strain evolved. And then here, these ones share more, they are more closely related than, say, the ones that are on this branch. But and then the scale here tells you how many substitutions are occurring per a particular position in the genome per year. So with all this information, you can really learn a lot about the pathogen, how it is spreading, how many people it is affecting. The values here are what we call support values. That tells you the confidence the software puts on this particular topology of the tree. 1 is the strongest, and so it varies. Sometimes we can also record it as percentages, so then it will be like 100% and all that. But we have to always be cautious when we are interpreting phylogenetic trees, because if the population is not well sampled, we can mislead ourselves. Because, for example, here we're looking at the past to the present, and each of these branches here have, if you have a fully sampled population, you can see that this here, it represents introductions of new strains or new variants into the population. However, with just one sequence missing, you can't see the other introductions, and you might be tempted to think, oh, there was only one introduction here, you see. And the more we miss data, the more we don't fully see the picture of the introductions. And here you completely would think that the orange one is the most, divergent, but this may actually be where your introductions are. So it's very important that we are cautious when we interpret phylogenetic trees, but there are software and computational ways to work around the difficulty with sampling. So here I'm showing you one. I'm now going into the cases, so we're almost to the end. But here I'm showing you what happened during the first Ebola outbreak in Sierra Leone. Data was gathered. There had been a prior study that looked at just 3 sequences that came from Guinea. And what was realized was that the early infections in Sierra Leone shared some similarities with the genomes that came from the Guinea. And you can see the Guinea tree lineages at the top here. And one of those lineages seem to share some mutations with what we call SL1 strain of the Ebola virus that started the outbreak in Sierra Leone. There were 2 to 5 mutations that were shared by this with the Guinean genome. Now, that SL1 seemed to have also diverged at a point, and it started gathering its own different mutations. Now, the speculation is that the SL1 was probably in the population several months before it was actually detected. That's a little bit interesting, because if you consider the way Ebola acts, you could, but this is what the data seems to suggest. So SL2 has 4 additional mutations that suggest that it has evolved from SL1. And then that further diverged into this SL3, which became the dominant one, completely wiping out SL2 in the population during the outbreak. Now, some of these green lines you see here are foreign doctors who came to Sierra Leone to help, but they acquired the virus and they traveled to UK and Germany and other places, and they were cared for. So here's the thing. You can see that with this haplotype tree, the SL3 became the dominant one, later on diverging into SL4 and running the whole outbreak in Sierra Leone. But to appreciate this story even more, you would have to go back a little bit and sample the population much more detail. Here is a study that followed the previous one that I just showed you, but they went in to gather a lot more data, about 1,600 data, and they gathered it across the 3 countries that the outbreak occurred in. So Guinea, Sierra Leone, and Liberia, covering a period of 2013 to 2016. They were able to use genomic data to pinpoint the origin of the initial outbreak in Sierra Leone to this, they call it prefecture in Guinea, that is here in this blast zone here. And it is called, I don't know what this name is pronounced, but something like that. Now, from there seem to have been dispersal events coming into other parts of Guinea, other parts of Liberia, and even other parts of Sierra Leone. So you can see this is the tree. This here represents time to the most recent ancestor, and this is the backbone. This seems to be the root, and it's spread in Guinea. All these lineages were within Guinea, and then for whatever reason, this branch here came on and started spread, and somebody brought it to Sierra Leone. And then we see another one here diverging to spread in Guinea, and then it went on and on. Here is the Liberia one that got spread to Liberia. So as the pandemic went on, as the epidemic went on, the countries decided to close their borders. So the first line that you see here, this purple or blue, whatever color this is, was the first time the Sierra Leone government decided to close their border. But before they closed it, by the time they closed it was too late, because you see this Kedo strain, it had already come from Guinea and was already here. The green color here shows that this is the origin that it came from. It was already in the country and was already spreading and starting to establish clusters in the community. Even after the closure, there were still variant transmission between the countries. Guinea was still receiving strains, because you can see these colors here that are coming from Sierra Leone. And when Liberia got hit as well, they closed their border around this time, and then Guinea closed their border late. But you can see the border closure didn't really do much. And that tells us something. We've seen this before. During the SARS-CoV-2 pandemic, most countries thought that by closing their borders, they would limit the spread or the importation in their country. Well, I don't know if it worked, because we can all attest to the fact that there were still imported cases. And for most cases, these already were in the countries before the borders were closed. So again, using genomic data, they were able to track the different clusters that came in. For example, if you take this particular strain, it came from Sierra Leone, Kenema, and it came to establish its own cluster, completely different from what was already in Guinea. And you can see similar events in different parts of the countries as the epidemic went on. And this is all possible through genomic data. So in summary, for this Ebola case, I would say that despite the fact that the border closure may have stopped the dispersion across borders, it wasn't very effective, in my opinion. And it has never really shown to be very effective, given the COVID experience as well. And we also see that the neighboring, there's more data in the paper that I haven't shown, but the neighboring countries were equally more susceptible. So countries like Cote d'Ivoire and others could have easily had it, but they didn't get a lot of introductions into their country. And the nature of the epidemic was heterogeneous, and that really helps to plan for future epidemics in these areas. Now, let's talk a little bit about COVID, because COVID is now something that we can't talk about genomic epidemiology without. COVID, this is the current picture globally. This data was just accessed last night, and you can see that with genomic data, we have been able to track the evolution of different variants of the virus almost across the entire globe. And currently, these are the variants that are still causing outbreaks in different parts of the country, of the globe. These are the EG5 and EG5 star strains or variants. And if you look at the Venn diagrams here, pie charts here, sorry, so far in Africa, these don't seem to be present. We don't know whether it is because we are not sampling as we used to, or it is not really here. But I mean, South Africa, for example, does a very good job sampling, but for it, we haven't seen it here yet. And so this still tells you that there is a need to keep an eye out. It seems to be in Asia and somewhere in the southern part of Asia, and also somewhere in Europe and America, Canada and the US. But so far, we haven't yet seen it. A little bit probably in this part of South America, but we haven't yet seen it in Africa. So we should keep an eye out on that. Now, a lot happened during COVID. So that was the world. But in Africa, all the African genomes were put together in this very beautiful paper by Tegali et al. And they here show the main variants of concern or variant of interest were over a period of time. And here was 2019 2019 was the very beginning. So we're looking at it from the very origin up to 2021. These here were highlighted because at the time, they were either variant of concern or variant of interest. And this is the distribution across. This is how they kind of evolved across. We could see clearly that a lot of, in fact, all of them were imported. So the black bars here shows you that these were imported from African countries to African countries, and from African country to African country. And the white bars here shows you proportion of the variants that were imported from outside of Africa. So a lot of our infections at the beginning were from outside, but later on, the importations between and the countries were within were from other African countries. And by absolute numbers, you can see, well, as far as importation from outside of Africa was concerned, Europe was the main origin of most of the variants that were being reported in Africa. And here is an alluvial plot that gives you an indication of the direction of the spread. So on the left here, you have the origin, and on the right here, you have the destination. And so if you take, for example, South Africa, there were a lot of spread from South Africa to other parts of Africa and to other parts of the world. You could say that this may be because South Africa was effectively doing the sampling. If other countries may have done sampling, they may have ended up with a similar picture. Also, we had the rest of the world spreading a lot of the variants to other parts of Africa. And then, of course, we have some African countries also spreading some to the rest of the world. So this is a very nice way of showing, using genomic data, the direction of transmission in the population. So we've talked about the world, we've talked about Africa. What was happening home here in Ghana? In Ghana, the very early sequences were generated, and we saw that they were very close. These were covering a couple of months, but you could clearly see that they were quite closely related. If you take the Wuhan strain at this time, and all the Ghana strains, we did 2 batches of sequencing. The first one was this yellow. So everywhere you see a yellow dot, it comes from the first batch of sequencing. And everywhere you see a blue dot comes from the second batch of sequencing. And you can see that they were quite closely related. Some of the second batches had a few more mutations than some of the first batches, but it's not always the case. There were some of the first batches here that were having more mutations than the second batches. So it kind of had a mixture of things, but we had already spotted that there were evidence that the virus was changing even within the Ghanaian population. This was published and it's available in Ngoi et al. There was another study by from Prof. Awandari's team in WACBIP, and they looked at the entire country. And so you can see clearly, and they also have some imported cases, Moranga et al. And you can see that we imported cases, obviously, but Greater Accra seems to be the origin, the place that was hardest hit, but also Central and Ashanti. And again, this shows you the variants that were gathered during the sampling. At the time of this paper, majority was the Delta, followed by the Alpha. And then, of course, we had this B11318 that was also spreading quite significantly. It never became a variant of concern for whatever reason. So you look at that, and this here shows you that by region, the proportion of the sequences. And again, there's not too much to tell from this, but you can see that the different, these are variants that were not classified as variants of concern, but were of interest. And so you see the B1, for example, being one of the main variants that was in the different regions. And if you look again at this place in 2021, this was at the very early stages, but in 2021, you could see the shift in the distribution of the variant. We still had a bit of B1, but it was almost being drowned out by the Alpha and the Delta, which was beginning to take root. We had another study by Dr. Ivy Asante and team and Professor William Ampofo from Noguchi here. They also looked at the entire country. What they did was really nice because they were leveraging on the influenza surveillance network, and they had data for both influenza and COVID, and they were looking at this side by side. But this was more focused here on COVID. And you could clearly see that they were able to capture, using genomic data, the different waves that occurred. The first wave was driven by all these other variants, which were none of these other colorful ones you see down here. And this was typical. We saw this even in the earlier paper that was published in Ngoi et al. 2020. You could see that at the beginning of the outbreak in Ghana, there were so many different variants that were driving the outbreak until a dominant one like Alpha emerged and completely took over. And then that established the second wave, and the third wave by Delta, clearly seen here. And this was just the beginning of the Omicron wave, which still persists as we speak. That paper also showed other things about travel. So this black bar here shows you the travel, and then you have the other lineages in this here, and it again almost similar information, but telling you other parts of the country where this was spreading. That's the last bar here, and you can see that majority of the cases were spreading, were picked from Central. They were coming from Central America. This is the travelers data, and this one here is the local, the Ghana data, with a few international samples that were collected here. And the distribution of variants was not very different, except this particular one here. But you can see that almost the same thing that was being imported was what was established in our population. So this was at the country level. Then we zoomed in even closer, because when you look at the Ghana data, this is coming from the Ghana Health Service website as at 2022 February. You could see that throughout the country, about almost 60% of our cases were in the Greater Accra region. So we did another study where we were looking for the way the disease was spreading within Greater Accra, the districts of Greater Accra region. And so here was what we found. This is the distribution of every dot you see here is the green ones represent the Delta variant. This one represents the Alpha, and here we have the Eta, and this was the Omicron, just when the Omicron was starting. So you can see that a lot of our lineages were coming from the Greater Accra or the Accra metropolitan area, and a few spread across other districts within the Greater Accra region. We also had a few data from international travelers, and we looked at that. I will talk a bit about that in a second. But this was what was happening with time. So when you look at it at the beginning, where unfortunately, some of the data that were coming in were not properly characterized as to which district they were coming from. So in the paper, we just called them unnamed district, but we know they were coming from Greater Accra. And so you could see that a lot of the Alpha and Eta lineages were all from these other districts. But with time, the Accra metropolitan area became the dominant, the area of the most spread, and kind of dominating the spread within the Greater Accra region. The other districts in Greater Accra saw very, very little of the spread. It could be also that the sampling wasn't as thorough in those places, and that is why we don't have much data. But we did a similar thing with the alluvial plot, and here you can see, with adding data from other parts of Africa and other parts of the world, you can see that a chunk, this is again the origin, and this is the destination. A chunk of the spread was coming from the Greater Accra, the Accra metropolitan area to other parts of Accra, other districts, or even the unnamed district that we couldn't find. A chunk of the spread was coming from here, as well as even to outside of Ghana. And again, these are the absolute numbers. You can see how many. Here was the importation. We kind of received a lot of imports from other African countries and then also from these other districts. Here we're looking specifically at the Accra metropolitan area. Where were the variants that were found there? Where were they coming from? And also, where were they going? So here we can clearly see that some were coming from other districts, some were coming from other parts of Africa, and so on, and a few from Asia. And where were they also being sent to? They were mainly being sent to other districts within the Greater Accra region. And it would have been nice to have data from other parts of Ghana to see whether these were also going as far as that. We also exported a few to Asia and other parts of North America. This will be the last one I talk about. The recent Marburg outbreak. This was by Professor Kofi Bonny from the Virology Department of Noguchi, and the NAMRU team, as well as the other people that are collaborating across Africa. And here we were able to use genomic sequencing to pinpoint the closeness of the Ghana outbreak to this Guinea outbreak or this Guinea strain here. Again, using the same concept of phylogeny, we could see that this is where, and the two samples from Ghana were very closely related. That's why they are all sitting on the same branch. And this may be because one infected the other or something, or they all got it from a common source. We don't know yet. I mean, the epidemiological data can help establish this. But with genomic data, we see a very strong relationship between these two, and also with the Guinea variant or strain. So a few concluding thoughts. Genomic sequencing data has shed light on the dynamics of transmission of diseases in different outbreaks. And you could see that using this, we could easily characterize the organisms, and this can easily help to either develop interventions such as vaccines or diagnostics, or help with deployment of intervention measures and all other kind of interventions that can be used to minimize the spread of these infectious agents. And we also realize that the border closures are often not very helpful. I'm not advocating for a loose border, but if we are going to close borders, we should close them and make sure that they are tightly closed. And we should close them much, much earlier, not wait till the pathogens have been imported before we close. If we can come to that agreement, I think it will be effective. But as it stands now, it hasn't been very effective in any of the outbreaks we have seen so far. Effective collaboration is extremely key between both local and international partners when there are disease outbreaks, and also rapid data sharing. There's been a lot of data shared on GISET since the outbreak of SARS-CoV-2. Currently, there are over 16 million SARS-CoV-2 genomes in GISET, and as part of this, it raises a lot of questions about data sharing. And sometimes one thing that I would say has helped a lot, we've changed a lot since the Ebola outbreak, because people like Professor Sabeti from Broad Institute and others, they, at the very early stages of Ebola in Sierra Leone, they realized that it would be more helpful to share data before publishing. So most of the data we saw were already available in the public domain before they were published in scientific papers. And I think a similar thing has been helpful in the COVID response as well. But we have to put the right structures in place. And so together at the Africa CDC team, there's a paper on a Pan-African genomic data sharing platform that is seeking to address some of these things. And I think when it comes live and we all support it, it will help us so that we can have equity as far as data sharing is concerned. Because if we share data and it's used to develop interventions and we don't benefit from them, then what is the use in that data sharing? Or if we share data and we become stigmatized and we can't travel to other places, like what happened in South Africa, then it doesn't encourage transparency and data sharing. So here are a few recommendations. This was by courtesy of one of my research assistants, Emeritus Professor Bennis, and I really acknowledge her for this. So investment, she recommends we invest, and I completely agree with her. We have to encourage our governments to invest in genomic architecture and the bioinformatic infrastructure that is required for effective genomic surveillance. We also have to promote data sharing, but within ethical boundaries and within equity. And we need to establish strong governance around data sharing. And again, we must expand our research. Our research should not just go with only identifying variants and leaving it there. We in this part of the world must begin to look into downstream translational research with the genomic data we generate to be able to develop diagnostics. Because if there's an outbreak here and we have isolated and sequenced the organism, we should be able to quickly develop rapid diagnostic tools or some kind of vaccine or some kind of immunotherapy or to inform drug design to be able to have a better response to the outbreak. With that, I want to say a big thank you to everyone, to the team for inviting me to do this talk, and also to my research assistants and everyone. Thank you for your attention. I'll be happy to take some of the questions you have. All right. Over to you, Professor Bisi.
Yes, thank you very much, Dr. Edu. This has been very enlightening, very interesting insights as to how we can apply genomics to disease surveillance, and also even for taking us through the various types of sequencing platforms that are available. So thank you very much. This is one that I think will go a long way to help all of us. There are a few questions in the chat. I'm going to take them one by one, but if anybody also has a question, you can ask. But I just want to, before we go to Q&A, mention the fact that this is a joint activity that is being presented both on the COBREG and then the ISG platforms to our members. Currently, we also have a workshop that is ongoing in ELISA. We're having a practical workshop in ELISA. I don't know if we can see our colleagues who are in that ELISA workshop. They are also joining us on this feed to also listen to this talk. So this is very, very good presentation that you have delivered for us. If there are any questions, you can please raise your hand, and then we can give you the chance to ask your question. But before we get there, I will start by looking at the questions that are in the chat. So we have three as of now. I think one is a comment and more or less an intervention. So there's someone saying, Dr. Edu, great presentation, but I saw in one of your tables, 1 to 11 days for a run on Illumina instrument. There must be a typo here because Illumina doesn't have runs that last 11 days. And says it takes less than 19 hours to run. So
It's
that's
obviously a typo. That one is quick.
Yeah.
Sorry about that, Rudolph.
Okay.
Yes. There is a typo
Yes.
there. None of these platforms, your platforms run 11 days.
11 days, yes. Okay.
No.
Okay, so there is another question from David Kwate. He says, hi, Bright. Thanks for this talk, important storytelling and highlighting our capabilities as scientists. Do you know how transferable gene sequencing is to reproduce proteins that can be used as vaccine markers? He's wondering if there is an error rate similar to natural mutations.
If I understand the question, I think you're asking how we can translate genome sequencing data to vaccines or as vaccine targets. If that is it, then I think that it's quite. I mean, we have a team here, for example, Professor Linda Amoa, who has capabilities or the capacity to express proteins for vaccine development. So that is quite doable, and usually you may have to do some codon optimization or something to allow it to express in the kind of system you are using, but it is something that can be done and it's doable. Yeah.
Okay, so Dr. Edu, if you can briefly stop sharing, we'll go on with the Q&A, but just briefly stop sharing so that we see our participants. So the next question is from Francis Badamusi, who asks, please, can we get the recorded video up? Yeah. So for all the COBREG webinars, you get the recording on our YouTube channel, and these will also be uploaded to the website for those who want to view again to look at. So you get it in two places, on the COBREG website, but you also get it on YouTube, all the videos of the webinars that COBREG has held. There's another question. This is an anonymous person. It says, please, is sampling in Ghana and Africa a major problem to accurate and effective research, and what can be done about it?
Yeah, I mean, you're perfectly right. Sampling is a big challenge, even within a small space like Ghana. Sometimes you receive samples and you are a bit unsure whether, I mean, the person is a male or female. You can understand male or female. The person is sitting in front of you or something. So that kind of information should not be too difficult to capture. But we get messiness in metadata like this, and it creates problems. The actual design of our sampling strategies in response to outbreak also needs to be critically looked at, because you may be oversampling from a particular population and undersampling elsewhere. So that's why when outbreaks like this happen, we need to have a team that is really looking at our design. How do we go about designing our sampling strategy to capture all the areas that need to be captured so that we can have a representative sampling? I have seen places like South Africa that seem to have this working very, very nicely. At least for COVID, there were a couple of instances where I listened to talks that they seem to have a very well-structured way of getting samples so that it's representative. And I hope that that kind of thing can be replicated in other parts of Africa and also in Ghana. But you're right, it's a definite challenge that needs to be dealt with more seriously.
Okay, thank you very much. So we now go to live questions. So Frederica, your hand is up if you can ask a question.
Yes, thank you, Dr. Edu, for the talk. I'm just wondering, so it looks like the imported variants are able to establish themselves much more than the locally generated variants. Do you know why that seems to happen? Because it looks like with each wave we've experienced, it's as a result of the imported variants. Although there are emerging variants locally, they are not able to sort of establish themselves so well.
Yeah. Well, it's a very difficult question to answer with any degree of certainty, but I could speculate. If a variant has established itself in one population, it must be doing something right. It must be having some advantages that allows it to perhaps replicate that same success in other populations. And therefore, it's not too surprising to me that because remember, variants are constantly being filtered out in populations. It is only variants that have a very advantageous trait, very, very strong advantageous traits that get established to the extent that they can actually be transmitted to other places. And so if it has already won, so to say, in quote, if it has already won the battle in another population, in other populations, coming into a different population becomes a little bit more easier. Also, it may be coming to meet an existing immune system that is quite new. And before the immune system catches up, by that time, maybe it has already established itself. That may be one of the reasons as well. But if we consider what happened with, let's say, Omicron, or even Delta, when Delta came, it quickly took over the whole thing and it wiped out Alpha completely. Alpha was around before Delta came. But for whatever reason, the fitness of Delta was simply too much for Alpha to coexist with it. And therefore, almost all the infections that we saw were of Delta. And then we thought Delta was the strongest until Omicron came and completely wiped out, so anytime a variant kind of establishes itself in one population and it gets into a new population, it seems to adopt almost the same kind of advantages and therefore be able to take over the transmission. It was a similar thing with the Ebola as well in South Africa, in Sierra Leone.
Okay, thank you very much. So next, Eric has his hand up. Eric, if you would want to ask a question.
Yes, one of the participants wants to ask a question.
Okay. So Eric is handling the ELISA workshop now. So we wanted to show them, but we're having some challenges. But we can work with this now. So please mention your name and go ahead with your question.
All right, Doc, thank you very much for your presentation. My name is Salom. I wanted to know, can we use sequencing in personalized medicine, where, for example, individual genetic profiles are analyzed for more better and targeted intervention instead of the usual wholesale interventions that, the usual wholesale interventions?
Yeah, thank you, Salom. I can tell you that yes, and it's already being done. In other places, people have their genomes sequenced, and these genomes are being used to manage them, for example, when they are sick. And it's also being used to inform their health in so many ways. So yes, this is actually the future, and it's not too far a future. I think a lot of people are buying into the idea of one drug fits all doesn't really work. We know, for example, some people have different copy numbers of some of these drug metabolizing enzymes. And that makes it they are able to quickly break down the drugs that ordinarily should take maybe 6 hours in their blood. It may end up taking 2 hours or 3 hours. And the doctor has administered the drug and he's thinking the next dose should be administered after so and so hours. But the person's genome says otherwise. The genome is completely different than what you would normally expect. So personalized medicine is something that is really taking on very strongly, and a lot of people are buying into that idea. And with the cost of genome sequencing becoming cheaper and cheaper and cheaper, it's actually going to be the way to go. So it's happening, and it's going to increase in its usage with time.
Okay, thank you very much. So I go back to the chat, the Q&A window. So the next question is from an anonymous person. He says, are you able to tell the success rate of application of genomics to public health in Ghana?
If you say success rates, meaning that the application of genomics stop an outbreak or something, that would be difficult to estimate. But what I can tell you is that genome sequencing has been very useful in informing decisions. For example, how did we know that a particular variant was in Ghana? It was through genome sequencing. So the government could come out and announce that this variant is here, people should be careful, and people should not do this, people should do that. How did we know which vaccines to go for? It is still through genome sequencing for us to know that, oh, this particular vaccine is not efficacious against maybe a particular variant of the virus, or we shouldn't use it. We can use almost the same kind of analogy for diagnostics. Malaria, for example, HRP2, HRP3 deletion, if we can do some kind of sequencing or some typing, we can be able to tell that this particular kit is not useful. So we shouldn't use. So it is useful in so many ways, but genomics alone as a tool cannot block or prevent an outbreak from coming. What it can do is that it can help to minimize the spread, or it can help to curb it, and so that it doesn't go far or spread very quickly, because we will get useful information to make decisions. Over.
Okay. Yeah, thank you very much. So I read the next question. It says, with regards to the efficient information sharing, do we have a national data bank where locally sequenced genomes can be accessed?
I have to be honest, I really don't know. I don't think as a country we have a repository for genomes. But this is a good question, because perhaps it is about time. We are sequencing a lot in this country nowadays. Bacteria genomes, viral genomes, parasite genomes, even human genomes. So I think it's about time that conversation is had, and we as a nation decide to do. But again, it is because such things do not exist in this part of the world. That is why the Africa CDC is proposing a framework for pathogen genomic data sharing. And the hope is that it may be escalated to include other, like human genomes or other things. Yeah.
Okay.
A very good one.
Yeah. So I have two quick questions in the chat, and then
Okay.
come back to the live platform. So the next one says, can the high rate of SARS-CoV-2 in Greater Accra also be associated with the high rate of travelers coming to Ghana from that point, and also the population density of Greater Accra?
Yes, certainly. I mean, we looked a little bit into that. For example, Accra, anywhere you have a very high population density, often outbreaks can spread very quickly. So the reason you could see clearly that was the area where most of the infections were coming from. And it's probably because of the closeness of human to human in trade, for example. If you take the Accra metropolitan area, it is among the busiest parts of Ghana. So it makes a lot of sense that people are coming into contact with each other, and therefore that is why they are getting infected. Also, the airport, for example, is right here, and most travelers are coming in, and sometimes we get able to catch them when they are at the port, but other times they sneak through, and it's not because they are mischievous or anything, but the disease may not have been at the level where we can easily detect, or the detection tool may lack sensitivity at the stage of the disease when they came. So several reasons may have caused them to sneak through, and therefore the outbreak could easily be established through those.
Okay. All right. Thank you very much. I'll take the last one, then we'll go to the live session. So the next one says, are there any agencies in Ghana that work on this personalized medicine? I think in relation to the earlier question.
Yeah, in relation to the first question. So again, I have to say, frankly, I don't know. But at Noguchi, I know that we are beginning to discuss this. The director is very interested in some form of, what do we call it? Genetic medicine or genomic medicine. We actually have Professor Oferiakwe's
the Center of Excellence.
Center of Excellence, one of the ACEs by the World Bank. It's a genomic medicine center. So already you can see that there is so much interest in this, even within Ghana here. And I think that it's almost similar in other parts of Africa. What we need is the proper legal framework, because, when you sequence somebody's genome, it is quite a lot of data to have on a person. It is a lot. And you want to be able to protect that data correctly. You want to be able to know who has access to the data and what is it being used for. All those things must be in place. If you sequence somebody's data and you discover that the person has a certain high risk of, let's say, some disease, some terminal disease, what do you do? So, or if, let's say, for a carrier sequencing, two people are about to get married, you sequence their genomes, and you discover that if they get married, they're going to have a child with cystic fibrosis or something. What are you going to tell them? These two lovebirds, are you going to separate them? I mean, it's a similar thing with sickle cell, but there's so many implications and so many things to really deal with so that we are prepared for it. But it is coming, and I believe people are really interested in it in Ghana here. So,
yeah. Great. Thank you very much. Eric, your hand was up. Do you have another question from the floor?
No, I just wanted to add that, Africa CDC is also sponsoring, based on the question I was asked, Africa CDC is also sponsoring this biobanking idea, where data and samples to be collected across Africa, and then those samples to be shared amongst Africans. So we are gearing towards that.
Okay, that's good. Thank you for information. So there are 3 more questions in the Q&A, but they are very long questions. So I'll try and quickly summarize. First one from Christopher Doku says, nice presentation. Please, can you tell us how Ghana is preparing in terms of capacity building in areas such as genomics, bioinformatics, etc.? For instance, how is our tertiary institution positioning our curriculum to enable smooth transitioning into other fields with different backgrounds?
Okay, so I mean, I can say that since COVID, almost every part of Africa, I see capacity building in genomics and bioinformatics. The Africa CDC, the WHO, with partners like Illumina, Nanopore, everybody is trying to build capacity in Africa for genomic sequencing. So it is coming. Bioinformatics is also following closely. We need the right infrastructure to support high computational genomics. But all that, I believe, is a process. And I think it has begun and it is starting. As far as curriculum is concerned, I know that there are curriculum in KNUST for bioinformatics, for example. And there are also some aspects of that in some programs in the University of Ghana. And so it is coming up. It is coming up. 3 years ago or 4 years ago, I don't think we had this kind of attention as far as genomics and bioinformatics are concerned. But with COVID, there's been so much attention on these that I can't imagine why an institution would not want to develop a curriculum in these areas. It will just be left out or left behind. So it's coming up. Yeah.
Okay, thank you. The next question is in two parts, but I think the first one has been touched on. It's talking about expansion of genomics, especially in resource-limited settings, and application of personalized medicine in the role of genetic studies. But the second part of that question is, what do you recommend in translating data obtained from genomic studies or candidate gene studies in intervention development, such as diagnosis?
Yep. I think that was one of the recommendations that I made. I think that we have to, you see, we need a plan, we need a strategy that allows us to move into translation after the basic science is done. Because it's not enough, in my opinion, and I believe you agree, that's why you're asking this question. It's not enough to say, here is the organism, we've sequenced it, and here is the phylogenetic tree. It helps, it's good, it's very useful. But how do we then actually use that information to develop some countermeasure? So the drugs, and we need to build capacity in translational research. If it's a vaccine that we want to make, we have the genome. We can identify the genes. We can look for those that are those that may be interacting with the immune system and are likely to elicit immune responses. We can make candidate antigens to those, clone them, express them, and then make some kind of animal studies to test the efficacy of these constructs. We can develop some T-cell based vaccines. We can do many things. Once we know the genome, a lot can be done. And I think that there's already interest in that. Even at Noguchi here, people are very much interested in developing these.
Okay, thank you very much. So next we have from an anonymous person. He says, do you believe that government funding for genetic surveillance and health research has improved significantly since the COVID-19 pandemic? And do you think this trend is likely to continue in the years to come?
Okay, this question you're asking, you may want to put me in trouble. But I think it's fair to say that the government did support genomic surveillance activities during COVID, because, I mean, some funds were made available. So yes, the government did something. After COVID, I am not too sure what the situation is like, because it's not that we don't have other diseases that we should be doing genomic surveillance on. So I'm expecting that there will be sustained funding. If the government did something during COVID, it shouldn't end there, because who knows when the next pandemic will come. And it is the capacities we build now outside of pandemic that will help us to fight the next pandemic. We should never be in the situation we were in COVID pandemic, where even the reagents and the equipment and the support systems and we didn't have almost any, none of those was available. So now that we have we have a breather without COVID, we should we should prepare the grounds and build the capacity to be able to respond more adequately to pandemics and also to diseases that are already here. We have malaria with us, it still kills, you know. We have different bacterial infections, we have different viral infections that we still need to pay attention to. So the funding shouldn't end. I pray that there's some government person listening so that they will go back and hopefully bring in some more funds. But we do need a lot of funding to do this. We can't wait for outsiders to fund what is supposed to benefit our own population. We must do it ourselves.
Yeah, money is always something that is needed to make these things move. Thank you very much. I thought I had one last question. One has just popped up. So let me quickly go through. So how do we relate the genomic research done so far in Ghana on SARS-CoV-2 to the COVID vaccines in the market?
Well, I mean, almost all the sequences we generated in Ghana have also been shared with the public, and therefore any big pharma that is developing a vaccine is likely using our data in one form or the other. So our data has, I believe, contributed to telling the story, the global story of the disease, but it has also helped in the development of these interventions.
Yeah. So I think one other angle to that question is based on the variants you are seeing, are the vaccines being used still effective against the variants we are seeing in country?
Well, I mean, I would say yes, but we should keep in mind that I always say that Omicron was perhaps the biggest vaccine against COVID, or the most successful vaccine against COVID, because it appears the antibodies against Omicron have so strong a neutralizing activity against other variants that it seems to have been driving the thing. And the nice thing about, well, it's not nice, but in a way, the good thing is that not many people seem to have been experiencing very severe symptoms with Omicron. I mean, doesn't mean it doesn't kill. Depends on your immune state or your health status. If you have comorbidities or underlying conditions, it could be fatal. But for most of the population, I think Omicron has been a little bit more protective than,
Yeah, I agree perfectly with that. In fact, Omicron reached many people with whom we could not reach with a needle and the vaccine to vaccinate them naturally. It was really one that build people's immunities up over time. So I have one last question, and I hope it's a question. It says, would you like to know, that's from Jonas Lawson, would like to know how prepared is Ghana, Noguchi, and other medical research institutions towards any future outbreak?
I
Are
can
we
say
prepared?
that I can say that yes, we are prepared. There's more to be done, but we are prepared. We have more people trained in Ghana and at Noguchi doing genomics and bioinformatics than ever. We have built capacity in the infrastructure. We have through support from partners like Africa CDC, ASLM, Illumina, and Nanopore. We have built capacity in instrumentation. We have some minimal computational resources. So I think we are not there yet, but we are not where we used to be. And more can be done. But for what we have, and I know other institutions like WACBIB and across the country, UHAS, KNUST, that there's a lot of capacity building going on. And we are way more prepared today than we were in 2019 or early 2020.
Yeah, and there are indeed a number of private labs that have also undergone training for sequencing. Yeah. So, okay, the last question is related to that. So the person is asking, is Ghana, in Ghana, is there a plan to factor in genomic sequences in preparedness against future pandemics for other emerging and re-emerging pathogens?
Yes, I think the answer is yes. And they can't, I'm aware that there is a document being developed for genomic surveillance, for example, pathogen genomic surveillance in the country. So there's quite a bit going on behind the scenes. And because everybody agrees that you need to be ready for the next pandemic, and genomic surveillance is the key. I think almost every stakeholder agrees on this. So Ghana is preparing, I can say that.
Okay. All right. So on that very positive note, I think we've come to the end of this webinar. It's been an enlightening session. I think all the questions that have been asked have been answered appropriately. So thank you very much, Dr. Bright Edu. Dr. Edu is once again a senior research fellow with the Department of Immunology at the Noguchi Memorial Institute for Medical Research, University of Ghana. And he's also the coordinator for the Genomics Sequencing Hub at Noguchi Memorial Institute. So he has a lot of experience in sequencing and we have been very privileged to have him be a facilitator or resource person for this webinar. So thank you also to all of you who have been on. I think at peak I saw 132 people on. This has been a very well patronized webinar. So thank you all very much for making the time to join us. Also, thank you to those who have been working in the background to make this happen. You've all been very excellent. And like I said, we currently have an ISG COBREG ELISA workshop happening. So the participants in that, 24 of them, also joined this feed to listen to this webinar. And thank you to all those who have helped to put this together. Let me once again remind our members, ISG and COBREG both, that please, we need you to also make your dues payments, because we need our funds to run the associations and to make all of these that we are doing happen. So please, we will share the information once again. If you go for COBREG, especially if you go to the website, the information is also there on how to make those payments so that we can get the associations moving. So on this note, I'll thank Dr. Bright Edu again for making the time for us. I know he's very busy. I was actually very happy when he agreed to do this for us. And also thank everybody for making the time to be on the call today. Have a good day, good morning, good afternoon, good evening, wherever you are. And we will see you in January. We are in December, we will not be having a webinar. Our next webinar is likely to be in February or March. So we'll see you in the new year with more such activities for your benefit. So thank you all, and have a good day.
Thank you.
Yes.
Bye-bye.
Bye-bye.